Bismillahirrahmanirrahim. We start another MCQ paper. This is the June 2019 paper 1-3, and it has some very interesting questions in it, so I would like to discuss those with you all. Start with the question 1, which is again a very they usually start off with a very simple and an easy question. Which combination of lenses for a light microscope will give the greatest magnification? You know, the eyepiece lens, say, magnifies it 5 times, and the objective lens magnifies it 100 times. So 5 times 100 times, and this is 500. So 5 into 100 is 500, 10 into 40 is 400, and 15 into 40 is 600, and 15 into 100 is 1500. So the answer was D, which was the highest, the greatest magnification. Then coming to question 2, the photograph micrograph shows some mesophyll tissue from a dicotyledonous leaf. What is the magnification of the photomicrograph? Now what have you to do? You have to measure this. You have to measure this on the paper, and then you have to calculate whatever it is, however millimeter it is, then you convert it into micrometer, and then you divide it by 5. And because we have no method of measuring it here, so we can figure that out. And that, of course, would be, you would measure this 5 micrometer, and you would write the answer in millimeter, and then you would multiply it by 1000. Why would you multiply it by 1000? Because you want micrometer. And whatever the answer is, you multiply it with then 5. Why? Because here you have this 5 something micrometer, and then you divide by 5, and that is the magnification of the photomicrograph. Then coming to question 3. Now question 3 is a diagram shows a typical animal cell. You usually don't read that, as seen using an electron micrograph. Which of the numbered structures are needed for proteins to be secreted at the cell surface membrane? Now what is 1? 1 is the Golgi. What is 2? 2 is the RER. And what is 3 is the mitochondrion. And what is 4 is the SCR. And what is 5 is some sort of a lysosome. Looks like some sort of a vesicle. Now it says which of the numbered structures are needed for proteins to be secreted. So they have not said where to be made, because if they had to be made, then they would be RER, but no. To be secreted at the cell surface membranes. Okay, so it's made on the RER, went to the Golgi, and now, so it is this process. Number 1, this. First, of course, made of the RER, then gone to the Golgi, then secretory vesicles, and then finally out of the cell. So the answer to that was 1 and 3, which is the Golgi and mitochondrion, because it's an active process. What is this active process called? Exocytosis. Exit. Then you come to question number 4. Lysosomes vary in shape and size, making them difficult to identify. What describes a lysosome? First of all, it's a single phospholipid bilayer, so it's a single membrane. Mitochondria is a double membrane. Chloroplast is a double membrane, containing several different hydrolytic enzymes that may act inside or outside the cell. So a vesicle enclosed by a single membrane containing several different. Now this one is wrong. Why? Because it's controlled by a double membrane. Now that is a lot of BS, which is beautiful story. So this is wrong. Then a vesicle is surrounded by a single membrane found only in phagocytes. That's again wrong, because phagocytes are present in a whole lot of other cells as well. Why is this one wrong? Double membrane. So if you did not know, lysosomes is a single phospholipid bilayer, and it contains hydrolytic enzymes. Then it says question 5, which statements about ATP are correct? It is produced in mitochondria only. That's wrong. It can be hydrolyzed to form ADP. Yes, of course. ATP is broken down to ADP plus inorganic phosphate. So ATP broken down to ADP, and this is of course hydrolysis. Why? Because you need to add a water molecule, and the water molecule splits into H and OH. It can be hydrolyzed to form ADP, then it contains deoxyribose. No, it contains ribose sugar. The ATP molecule has a ribose sugar. It has an adenine base, that is why ATP, and this is a ribose sugar. And to this carbon 6 is attached 3 phosphates. So it contains deoxyribose. That's again a lot of BS. Beautiful story. It's ribose sugar. It's a ribose sugar, not a deoxyribose. That's a fact which you need to remember. Okay, next. Question 6. Question 6 says a single-celled organism lives inside the body of a fish. A single-celled organism lives inside the body of a fish. Fine, good. We have bacteria living inside us as well. This single-celled organism is rod-shaped, 700 micrometers long, enclosed in a cell wall, and has cytoplasm containing thousands of copies of small circular DNA plasmids. Which statement about this single-celled organism is correct? It is a eukaryote because it can be seen without using a microscope. It is a eukaryote because it is too large to be a prokaryote. It is plant because enclosed in a cell wall. Not necessarily. Prokaryotes have cell walls, fungus has cell walls, plant has cell walls, and certain protoctists have cell walls, algae. It is a prokaryote because it has a small circular DNA in its cytoplasm. Question 7. A solution of amylase was added to a suspension of starch. The mixture was stirred and kept at 40 degrees for 45 minutes. Samples were then tested for various reagents. What is the expected set of results? Iodine test. So first you added starch, and then you added amylase, and the amylase was going to convert, and you kept it at for 45 minutes. So all of this starch would be converted to maltose, and maltose is a reducing sugar. So the iodine test is black, but if starch is present, it becomes if the iodine is brown in color, but if you test it with starch, then it becomes blue-black to prove that starch is present. Now it remained brown, that means all the starch would have been used up, all the starch, so there'll be no starch left because all the amylase would have converted into maltose. Benedict's would be orange and the biuret test would be purple. Why? Because amylase is what? Amylase is a protein. And if you remember, protein purple present. Protein purple present. Now you would forget the amylase thing and you would not correlate it. Okay, oh my god, amylase is an enzyme. Enzymes are proteins. No, you didn't correlate it because you are not reading the question carefully and you're not concentrating properly. You have too much on your mind. Maybe watching too much of Netflix. Then we have question number 8. The diagram shows 3 hexose sugars. Which row correctly shows examples of carbohydrates in which these 3 hexose sugars occur? So you know what is what. So sucrose has 3, cellulose has 2. Why? Because it's beta glucose, and amylose has alpha glucose, so that is 1. So amylose and amylopectin are in starch, and that contains alpha glucose. Cellulose is made up of beta glucose, and sucrose has a glucose and a fructose. Then coming to question number 9. What can occur as a result of a condensation reaction? Condensation reaction, a disaccharide is formed, a glycosidic bond is broken, a molecule of water is produced, 2 monosaccharides joined together. Yeah, so it's 1, 3, and 4. Condensation means water exits. So what can occur as a result of a condensation reaction. You see, hydrolysis breaks bonds, but condensation forms bond. So this was wrong. This was the only thing that was wrong. And the answer was 1, 3, and 4. Then coming to question number 10, the diagram shows some relationships between features of carbohydrates. Alpha glucose is sort of a circle, and then 1,4 bonds and then branched. Which row correctly matches the carbohydrate with some of its features? Amylose, glycogen. So why was this the correct answer? You have to know this. So 1,4 bonds, alpha glucose. So what was 1 was amylose, and 2 was glycogen, and then of course amylopectin 3. Why? Because it contained all these, and then of course cellulose. Then coming to 11, which row describes a triglyceride? Which row describes a triglyceride? Hydrophilic and insoluble in alcohol. No, it is hydrophobic and it is soluble in alcohol. Then coming to question number 12. Now this question was I think a little confusing and a little difficult because it shows you 3 triglycerides X, Y, and Z. Now you look at this chain. I want you to concentrate at this chain. Now it's got no double bond. And then you go to this chain, this has no double bonds. And then you look at these chains, none of these have double bonds. So now when you look at the answer, why was the answer A? Because contains saturated fatty acids X, Y, and Z, contains unsaturated fatty acids X and Y, and contains more than 2 different fatty acids X and Y. So I mean, if you look at the question carefully, I'm sure you can figure it out why the answer was A and why were the others wrong. Then coming to the next question, now we come on to question number 14, 13, yeah. So in question number 13, which features of collagen result in it having a high tensile strength? Bonds form between adjacent molecules. Each three-stranded molecule is held together by hydrogen bonds. Every third amino acid in the polypeptide is small. The primary structure is held together by peptide bonds. So the important thing is that collagen is a fibrous protein. And of course, the answer to this was 1, 2, and 3. Why is 4 not in the picture? Why is 4 wrong? Why is 4 wrong is because you've got to understand it says which features of collagen result in having high tensile strength. Now the primary structure is present in every protein, but all proteins do not have high tensile strengths. So high tensile strength. Which features of collagen result in it having high tensile strength? This was the part which you had to really comprehend. So 1, 2, and 3, 1, 3, and 4, 1 and 2, 2, 3, and 4. So this was the answer to this was A, which is 1, 2, and 3, and that was the reason why that was the answer. Then coming to question 14, which levels of protein structure would always be changed if an enzyme works by the induced fit hypothesis? Would always be changed. So what we have to understand is protein structure would always be changed. You have to understand is what do we say? What is the induced fit hypothesis? The enzyme molds around the substrate. So if the enzyme molds around the substrate, then primary structure is not going to change, secondary is not going to change, the tertiary will only change because it only the 3D structure adapts a little to the substrate. So the answer was D. Then 15. Now when you look at question 15, it says the enzyme beta-galactosidase can catalyze the hydrolysis of 4 substances A, B, C, and D with similar structures. Each substrate has a different Km value. For which substrate does beta-glucose have the lowest affinity? So we know it's inverse. Lower the Km, higher the affinity. Higher the Km, lower the affinity. So that was A basic fact which you had to understand is lowest affinity would be the higher Km. So higher the Km, lower the affinity, or if you say the inverse, so that was the answer to that. Then coming to question 16, which is correct for competitive inhibitors of enzymes? They occupy the active site of an enzyme. They have exactly the same shape as the substrate. No, they don't have exactly the same shape. There's sort of, maybe a little bit of it could be the shape of the substrate. They can be used to control the rate of enzyme activity. Competitive, yes, can reduce the rate of activity. They can bind to a site of an enzyme other than the active site. No, I'm sorry. Competitive inhibitors have the same shape of the substrate, so they will always bind to the active site. They cannot bind to a site other than the active site. So that's why the answer was B. Then we come to question number 17. Now, if you look at question number 17, the diagram presents the fluid mosaic model of membrane structure. Which two of the numbered components contribute to the fluidity of the membrane? And the answer to this was 4 and 6. Now, 4 is cholesterol, and 6 is the fatty acid tails or the fatty acid chains. 1 is the phosphate head. 2 is some sort of a protein. 3 is glycolipids, glycoproteins. 5 is another protein, which is an intrinsic protein. And 6, of course, is the four fatty acid chain. So the answer was 4 and 6, cholesterol, and the fact that the fatty acid chains can be either saturated or unsaturated, and that, of course, will affect the fluidity of the membrane. Then we come on to question number 18. So question 18, if you look at question 18, the cell surface membrane of some cells are largely made up of phospholipids and cholesterol with few proteins. So which transport mechanisms will be reduced across these membranes? If there are no channel proteins, so active transport cannot take place. Facilitated diffusion requires channel protein, but it said few proteins. So which transport mechanism will be reduced across these membranes? Facilitated diffusion, active transport, or diffusion? So reduced will be 1 and 2 only. Why? Because diffusion is across the phospholipid bilayer that does not require a membrane protein. But the first two ones, which is facilitated diffusion and active transport, require channel proteins. So if channel proteins are not present, active transport will not occur. Then we come on to the question number 19. One of the glycoproteins present in the cell membrane of human cells is called ICAM1. ICAM1 is a cell adhesion molecule and is also used by the common cold virus to attach to human cells. Which statements are true about ICAM1? A drug binding to ICAM1 to block the common cold virus could stop cells attaching to each other. Part of the ICAM1 structure must be complementary to proteins in the coat of the common cold virus. The ICAM1 structure must have hydrophobic parts to stabilize it within the cell membrane. So, glycoproteins present in the cell membrane of human cell is called ICAM1. And this helps the cells to be held together. These are called cell adhesion molecules and is used by the common cold to attach. So which statements? And then of course it is all three of them were correct, which is 1, 2, and 3, and that's why the answer was A. As all of these contribute to the factor that they are telling us something about the ICAM1. Then question 20. A student weighed a cylinder of potato and then put it into a test tube containing a salt solution. The potato cylinder was removed from the salt solution after 1 hour. After 1 hour, it was removed, the potato cylinder. It was plotted dry and then reweighed. The student recorded that a potato had lost mass. So if it was, say, 2 grams, it became 1.5 grams. So which row shows the correct explanation of the results? You see, the water potential of the potato cell before soaking was higher. So condition of the potato cell after soaking was plasmolysed because it had lost the water. So it had become less turgid, or plasmolysed means it had become flaccid, or it had become softer, or it had lost its stiffness. So plasmolysed. Then we come on to question 21. Now, question 21 is some parts of a typical human chromosome are more numerous than others. Some parts. Which of the parts are listed in order from the most numerous to the least numerous? Most numerous to the least numerous. So, of course, we have histone, telomere, and DNA molecule, which would be, of course, the histone proteins are in large numbers, and then the telomeres are one on either side of the DNA. And the DNA molecules of every chromosome has how many DNA molecules? Two DNA molecules. So every chromosome has, so what you have to understand is it in order of the most numerous. So histone, telomere, and DNA molecule. Then number 22, the enzyme telomerase prevents loss of telomeres after many mitotic cell cycles. Which cells need to transcribe telomerase enzyme? Stem cells, activated memory B lymphocytes, helper T lymphocytes secreting cytokines. Now, of course, helper T cell secreting cytokines would not need to make telomerase because it makes this chemical. So those cells which are going to multiply will need the telomerase enzyme. So that was 1 and 2 only, because the memory B lymphocytes then multiply and form more and more copies of it. And the stem cells, of course, divide and then, of course, differentiate into different organs and tissues of the body. So that finishes this up till 22, and we'll continue this in the next video from 23 to 40. And thank you very much.